Hello students, today let us learn about plant and animal cells. Both plant cells and animal cells are eukaryotic cells and they share many similar characteristics including the presence of similar organelles. Unlike prokaryotic cells, the DNA in both plant and animal cells are present as chromatin threads within a nucleus that is enveloped by a nuclear membrane. In addition, both animal and plant cells contain other membrane-bound organelles that carry out specific functions. However, there are a number of differences between plant and animal cells. Plant cells can be differentiated from animal cells by their larger size, presence of cell wall, a large vacuole and plastids, since animal cells generally have irregular shapes, are without cell wall and also contain structures such as centrioles, lysosomes and cilia and flagella that are not found in plant cells. The characteristics of plant cells and animal cells are discussed here. First, let us see the characteristics of plant cells. Plant cells are generally larger than animal cells and are typically rectangular or cube shaped due to the presence of a rigid cell wall. A plant cell also contains a large vacuole and plastids in addition to other eukaryotic organelles. The general characteristic features of a typical plant cell are: first, cell wall. Plant cells contain an extracellular cell wall surrounding the plasma membrane. The cell wall is composed of cellulose, hemicellulose, pectin, while lignin is present in many cases where it is secreted by the protoplast on the outside of the cell membrane. Thus, the plant cell wall differs in composition from the cell walls of fungi and bacteria, which are made of chitin and peptidoglycan respectively. The cell wall is divided into the primary cell wall and the secondary cell wall. The primary cell wall is formed first and is extremely elastic, while the secondary cell wall is formed at the end of the cell growth. The cell wall plays an important function of the cell in controlling turgidity. The second one is the cell or plasma membrane. The plasma membrane of plant cells is a thin semi-permeable membrane that surrounds the cytoplasm of a cell enclosing its contents. The plasma membrane is characteristic of eukaryotic cells in containing carbohydrates attached to the proteins and sterols, as well as glycoproteins and glycolipids. Movement of materials across the plasma membrane occurs by passive and active processes as in prokaryotes. The third feature is the presence of plastids. The plastids are organelles bound by a double membrane present in plant cells. Chloroplasts are plastids which contain chlorophyll and the biochemical systems for light harvesting and photosynthesis and serve as the sites of photosynthesis in a plant cell. They contain their own DNA and 70S ribosomes. A typical plant cell contains as many as 50 chloroplasts. Other types of plastids present in plant cells are amyloplasts specialized for starch storage, eleoplasts specialized for fat storage, and chromoplasts specialized for synthesis and storage of pigments. The next part is the cytoplasm. Cytoplasm in plant cell consists of a gel-like fluid matrix surrounded by the cell membrane and contains water, enzymes, salts, organelles, and various organic molecules. The next feature are the cytoskeleton. A network of fibers representing the cytoskeleton is present throughout the cytoplasm of plant cells, and it helps in maintaining the shape of cell and gives support to the cell. The next characteristic is endoplasmic reticulum or ER. Endoplasmic reticulum or ER comprises of an extensive network of membranes composed of both regions with ribosomes, also called rough endoplasmic reticulum, and regions without ribosomes, also called smooth endoplasmic reticulum. The smooth endoplasmic reticulum plays a role in lipid biosynthesis, carbohydrate metabolism, and detoxification of toxic compounds, while protein synthesis and transport occurs on rough endoplasmic reticulum. The next is Golgi complex. The Golgi complex consists of flattened sacs of membranes called cisterns and functions in processing proteins and lipids through the addition of sugar molecules, producing glycoproteins or glycolipids that are used in membrane formation and cell-to-cell communication. Plant cells have many simple units of Golgi apparatus called dictyosomes. The next feature are the microtubules. Microtubules are hollow, rod-like structures present in the cells that function primarily to provide support and maintain the shape of cell. They also play a role in cytosol movement within the cell as well as during spindle formation for chromosome movement in mitosis and meiosis. The next is mitochondria. Mitochondria are the site of cellular respiration and ATP production. They have an outer membrane and an inner membrane with cristae, and the mitochondrial matrix contains the mitochondrial DNA, 70S ribosomes, and metabolic enzymes. They multiply by binary fission. The next component is the nucleus. The nucleus contains DNA in the form of chromosomes surrounded by a nuclear envelope, and the nuclear membrane has tiny holes called nucleopores that allows nucleic acids and proteins to move into and out of the nucleus. The nucleolus is located inside the nucleus of eukaryotic cells and serve as the site of ribosomal synthesis and the site for the first stages of ribosome assembly. The next feature is the presence of peroxisomes. Peroxisomes in plant cells are tiny vesicles bound by a single membrane that contain enzymes that produce hydrogen peroxide as a byproduct. These structures play a role in plant processes such as photorespiration. The next feature is the presence of plasmodesmata. Plant cells have characteristic pores or channels called plasmodesmata between the cell walls through which the plasma lemma and endoplasmic reticulum of adjacent cells are continuous that allow molecules and communication signals to pass between individual plant cells. Next is the ribosomes. Like all eukaryotic cells, plant cells contain 80S ribosomes attached to the rough endoplasmic reticulum or free in the cytoplasm, while 70S ribosomes are present in the chloroplast and mitochondria. Next is the vacuole. Vacuole is a large central water-filled vesicle enclosed by a membrane known as the tonoplast present in plant cells. The function of vacuole is to maintain the osmotic pressure and turgidity of the cell and also stores useful material and digests waste proteins and organelles. Let us now see the plant cell types. Plants are mostly multicellular and the cells of a plant body are of different types depending upon their shape, structure and function. The main types of plant cells are: the first type is the parenchyma cells. These are living cells with thin permeable primary walls and they have diverse functions including storage, secondary metabolite synthesis, support, photosynthesis and phloem loading. Parenchyma cells, primarily concerned with photosynthesis, contain many chloroplasts and are called chlorenchyma cells. The next type of cells are the collenchyma cells. Collenchyma cells are alive at maturity and have only a primary wall which is thickened only at the corners where 3 or more cells come in contact. Cell walls of collenchyma cells contain pectin and hemicellulose and lacks lignin. Collenchyma cells are typically elongated cells and divide transversely. The role of collenchyma cells is to give support to the plant and to confer flexibility and tensile strength on tissues. The third type of cells are the sclerenchyma cells. Sclerenchyma cells are hard and tough cells that develop an extensive secondary cell wall impregnated with lignin laid down on the inside of the primary cell wall. The secondary wall is hard and impermeable to water and thus sclerenchyma cells have an empty central cavity without cytoplasm and are typically dead at functional maturity. Their main function is to provide mechanical support. They are of two broad types: sclerates or stone cells and fibers. Let us now learn about the animal cells. Animal cells are also typical of the eukaryotic cell in being enclosed by a plasma membrane and containing a membrane-bound nucleus and organelles. Unlike plant cells, animal cells do not have a cell wall and animal cells come in various sizes and tend to have irregular shapes. Animal cells also differ from plant cells in containing structures such as centrioles, lysosomes and cilia and flagella that are not found in plant cells. The presence of specialized cells such as nerves and muscles in most groups except in some protozoans gave these organisms the ability to move about, which is lacking in their plant counterparts. Another unique characteristic of the animal kingdom is the presence of an extracellular matrix made of the protein collagen, which binds together the cells in most animal tissues, which is in contrast to plant and fungal tissues where cells are bound together by other molecules such as pectin. However, unlike plants which can carry out photosynthesis, animals are unable to manufacture their own food and therefore are always directly or indirectly dependent on plant life. The general characteristic features of a typical animal cells are: the first one is the centrosome and centrioles. They are small body located near the nucleus having a dense center with radiating tubules. The centrosomes serve as centers where microtubules are made. During mitosis, the centrosome divides into two parts and each part moves to the opposite sides of the dividing cell. The centrosomes in animal cells have centrioles. Centrioles are small self-replicating structures made up of 9 bundles of microtubules. They appear to help in organizing cell division but are considered not essential for the process. The next feature are the cilia and flagella. Cilia and flagella are commonly present in unicellular animals for locomotion, while in multicellular organisms they function to move fluid or materials past an immobile cell as well as in moving a cell or group of cells. The next feature is the plasma membrane, nucleus, endoplasmic reticulum and mitochondria. These organelles are characteristic of eukaryotic cells and are similar to those described in plant cells. The next is the Golgi apparatus. Animal cells have a single highly complex Golgi apparatus. The fifth characteristic is the ribosomes. Animal cells contain 80S ribosomes attached to the rough endoplasmic reticulum or free in the cytoplasm as in plant cells, while 70S ribosomes are present in mitochondria. The next feature is the endosomes and endocytosis. Most animal cells contain membrane-bound vesicles called endosomes, which are formed by the process of endocytosis involving invagination or folding inward of a cell's plasma membrane to surround macromolecules or other matter diffusing through the extracellular fluid. Next is the intermediate filaments. Intermediate filaments are a broad class of fibrous proteins which are 8 to 12 nanometers in size that function as structural elements of the cytoskeleton and play an important role in maintaining cell shape and rigidity. Next are the microfilaments. Microfilaments are solid rods made of globular proteins called actin, and they also form important component of the cytoskeleton. Next is the microtubules. Like all eukaryotic cells, animal cells also possess these straight hollow cylinders in their cytoplasm for transport and structural support. The next feature is lysosomes. Lysosomes are vesicles that contain the hydrolase enzymes to break down cellular waste products and debris from outside the cell. Next is peroxisomes. Peroxisomes are roughly spherical microbodies bound by a single membrane found in the cytoplasm, and they function to break down toxic substances like hydrogen peroxide or other metabolites. They are a major site of oxygen utilization and are numerous in the liver of animals where toxic byproducts accumulate. The next feature is the vacuoles. Vacuoles in animal cells are many and small. Let us now see the conclusion. Both plant cells and animal cells are eukaryotic cells and they have similar organelles. However, there are a number of differences between plant and animal cells. Plant cells are generally larger than animal cells, while animal cells come in various sizes and tend to have irregular shapes. Plant cells are more similar in size and are typically rectangular or cube shaped. Both plant and animal cells contain structures which are unique to each group and this differentiates them from one another. Plant cell contains cell wall, a large vacuole and plastids which support their autotrophic and sedentary nature, while the absence of cell wall and presence of cilia and flagella along with other specialized cells give mobility to animal cells. Thank you.